All right, ready to dive into some really cool stuff.
Yeah, let's do it.
Today we're talking about how our immune systems can actually fight cancer.
Sounds promising.
It is, and it all comes down to this molecule called CD86.
CD86. Okay. I've heard of it, but refreshing my memory.
Well, you sent me this article from Assay Genie titled CD86, Enhancing T-Cell Activation in Immunotherapy. It explains how we can actually use CD86 to boost cancer immunotherapy.
You're right. Immunotherapy is super hot right now, and this article really digs into why CD86 is so important.
So let's start with the basics. What exactly is CD86, and how does it help our immune system attack cancer cells?
Okay, so imagine our immune system is like a complex army, right?
I'm with you.
You've got your scouts, your warriors, and then you have a whole communication system to make sure everything is coordinated.
Makes sense.
CD86 is part of that communication system, a critical part.
So it's sending signals.
Yeah.
But what kinds of signals and to whom?
So CD86 is found on these cells called antigen-presenting cells, or APCs for short.
APCs, got it.
And these APCs, think of them like the scouts I mentioned before. They're constantly patrolling, looking for anything that looks suspicious, like a virus or, a cancer cell.
So they find the enemy. Then what?
Well, they need to tell the warriors, the T cells, to attack. And that's where CD86 comes in. It's like a bridge between those APCs and the T cells. It sends the signals that tell the T cells whether to attack or stand down.
So it can both activate and suppress the immune response.
Yeah, it's kind of two-faced like that.
Wow, that's interesting. How does that work?
Well, CD86 can interact with two main receptors on T cells, CD28 and CTLA4. When it binds to CD28, it's like giving the T cells a green light, full speed ahead, attack.
Amazing. But you said it can also suppress the immune response. Why would it do that?
Because, well, you don't want your immune system going haywire and attacking everything in sight, right?
Right. Friendly fire is bad.
Exactly. So that's where CTLA4 comes in. When CD86 binds to that receptor, it's more like a yellow light or even a red light. It tells the T cells to hold back, to chill out. It's a way of keeping the immune response in check.
So it's all about balance, making sure the immune system is strong enough to fight threats, but not so strong that it starts attacking the body itself.
That's the idea. It's a delicate balance. But here's the thing. Cancer cells are tricky. They've figured out how to mess with this system.
Oh no, how do they do that?
Well, think of it like this. Cancer cells are like hackers. They can actually hijack the CD86 CTLA4 pathway and make it work for them.
So they're tricking the immune system into thinking everything's fine when it's not.
Right. They're basically putting up a smokescreen, hiding from the immune system. And that's one of the reasons why some cancers are so tough to treat. They've created this whole environment that protects them from immune attack.
So they're using the body's own defenses against it. That's pretty sneaky. But that's where this research on CD86 gets really interesting, right?
Exactly. If we can understand how cancer cells are manipulating this system, we can find ways to fight back.
Okay, I'm ready for the next part of this story. Tell me more.
Well, let's talk about monoclonal antibodies. Specifically, this one called GL1.
I think I saw that mentioned in the article, GL1.
Yeah, and it's specifically designed to target CD86 and block its interaction with CTLA4.
So it's like taking away the cancer cell's ability to hit the brakes on the immune system.
That's a great way to put it. It's like, cutting the wires on the red light.
Okay, I like that analogy. So the T cells are no longer being suppressed.
Right. And not only that, but it actually enhances the signal from CD28, the green light, remember? So now it's like those T cells are getting a double dose of go get them.
This GL1 sounds pretty amazing.
It has a lot of potential.
But
And it's not just about the T cells either. Remember how we talked about that immunosuppressive environment that tumors create around themselves?
Yeah, that protective shield.
Well, GL1 can actually disrupt that too.
Wow, really? How so?
It all has to do with these other immune cells called TREGs, regulatory T cells.
Okay, I vaguely remember you mentioning those. What are they again?
You can think of them as kind of like peacekeepers, They're there to prevent the immune system from overreacting.
Right. But you said that the tumor can manipulate them?
Exactly. The tumor can actually trick those TREGs into helping it, making them part of that protective shield. But GL1 can actually interfere with that whole process.
So it's like taking down the tumor's defenses from the inside out.
Exactly. So we're unleashing the T cells, weakening the tumor's defenses, and potentially even turning some of the tumor's allies against it. It's pretty cool stuff.
Okay, I'm definitely starting to see why you're so excited about this. But I'm curious, are there any specific cancers where this approach might be particularly effective?
Actually, yes. The article specifically mentioned melanoma, non-small cell lung cancer, and colorectal cancer.
Okay, and what makes those cancers so vulnerable?
Well, they all seem to rely heavily on this CD86-mediated immunosuppression that we've been talking about. So targeting CD86 could really make a difference for those patients.
This is all incredibly fascinating. It really seems like CD86 could hold the key to some major breakthroughs in cancer treatment.
I think so too. But before we get ahead of ourselves, it's important to remember that there are still challenges to overcome.
Like what?
Well, like with any powerful tool, you have to be careful how you use it. One of the biggest concerns with immunotherapy is the risk of overstimulating the immune system.
Right, too much of a good thing can be bad.
Exactly. You want to boost the immune response, but you don't want to send it into overdrive. That could lead to autoimmune reactions where the immune system starts attacking healthy tissues.
So how do researchers deal with that risk?
Well, a lot of research is focused on figuring out who will benefit most from these treatments and who might be more likely to experience side effects. They're looking for what are called biomarkers, basically clues in a patient's body that can help predict how they'll respond to treatment.
So it's about personalizing the treatment, making sure it's the right approach for each individual.
Exactly. And that's what makes this whole field so exciting. The more we learn about the immune system and how it interacts with cancer, the better we'll get at developing these safe and effective therapies.
I'm ready to hear more.
All right, let's dive deeper into the specifics of GL1 and its potential in combination with other cancer therapies.
Sounds good to me. Welcome back. We're in the middle of talking about this fascinating molecule, CD86, and how it can be used to, well, manipulate the immune system, especially when it comes to cancer.
It's really powerful stuff. And, GL1, the monoclonal antibody we were talking about, it's really showing a lot of promise in this area.
Yeah, we were just getting to that before the break. Yeah. So GL1 is a monoclonal antibody, which means it's engineered to bind to a very specific target.
Exactly. And in this case, that target is CD86. Remember, CD86 is like that communication hub on the surface of immune cells.
Right, and it can send those stop signals to the T cells through CTLA-4.
Yep. And GL1 basically blocks that interaction. It's like, imagine it's a shield covering up that binding site on CD86 so CTLA-4 can't attach and deliver its message.
So the T cells are basically free to attack.
Not only that, but GL1 actually does something even cooler. It actually boosts the signal from CD28, the go signal.
So it's a double whammy. It's blocking the stop signal and amplifying the go signal at the same time.
You got it.
Yeah.
It's like giving those T cells a supercharge, making them way more effective at fighting the cancer.
And remember how we talked about those TREGs, the peacekeepers that tumors can manipulate? Does GL1 affect them too?
Oh, yeah, definitely. GL1 can actually interfere with the tumor's ability to, mess with those TREGs. It basically cuts off their supply line, so to speak.
So those TREGs can't help the tumor hide anymore.
Pretty much. It's like taking down the enemy's defenses from the inside.
Mm-hmm.
And all these different effects, they can really add up.
It's like a multi-pronged attack on cancer, right?
That's a great way to put it. And, some cancers are more vulnerable to this kind of approach than others.
The article mentioned melanoma, non-small cell lung cancer, and colorectal cancer.
Yeah, those are some of the big ones. They all tend to rely heavily on this CD86-mediated immunosuppression to survive. So blocking CD86 with something like GL1 could be a real game changer.
Okay, let's break that down a bit. Why those specific cancers? What makes them so, susceptible?
Well, take melanoma, for example. Melanoma cells often have really high levels of CD86 in their surroundings.
So there are a lot of targets for GL1 to latch onto.
Exactly. And early studies are already showing some really promising results. Some patients are seeing their tumors shrink, and they're feeling much better overall.
That's amazing. So for melanoma patients, this could be a real breakthrough.
It really could. And then you have non-small cell lung cancer. That one's interesting because it often involves these immune cells called tumor-associated macrophages, or TAMs for short.
TAMs, okay. Remind me, what are those again?
They're kind of like double agents in the immune system. Sometimes they help fight the tumor, and sometimes they actually end up helping it grow.
Oh, yeah, I remember. It's like they get tricked by the tumor.
Yeah, it's like a classic case of mistaken identity. But in lung cancer, a lot of these TAMs start expressing high levels of CD86.
So they're sending out those stop signals, even though they shouldn't be.
Right. But here's the cool part. GL1 can actually reprogram these TAMs, shifting them back towards their tumor-fighting role.
That's awesome. It's like taking another one of the tumor's weapons and turning it against them.
Exactly. And finally, we have colorectal cancer. This one's a little different because it's in the gut, and CD86 actually plays a really important role in regulating the immune response there.
So blocking CD86 in the gut, could that actually help prevent colorectal cancer from developing in the first place?
Potentially, yeah. But even if the cancer's already there, blocking CD86 could make the environment in the gut much less hospitable to those cancer cells, so it could help slow down the tumor's growth.
Wow, this GL1 really seems like it could be a valuable tool in the fight against all sorts of cancer. But the article also mentioned its potential in combination with other therapies.
Oh, yeah, that's another really exciting area. Because, cancer cells are clever. They often have multiple ways of evading the immune system. So using a combination of therapies can be much more effective than using any single therapy alone.
So it's like a 1-2 punch, hitting the cancer from multiple angles.
Exactly. For example, you could combine GL1 with another type of immunotherapy called a checkpoint inhibitor. Those work by targeting different checkpoint molecules like PD-1, which is another one of those stop signals.
So you're basically amplifying the go signal while also blocking multiple stop signals at the same time.
Exactly. It's like giving the immune system a real boost while also cutting off the tumor's escape routes.
That makes sense. But what about combining GL1 with chemotherapy? I thought chemo actually suppresses the immune system.
It can, yeah. And that's always been one of the big downsides of chemo. But there's some really interesting research showing that adding GL1 to the mix could actually help counteract that immunosuppression.
So it's like the chemo weakens the tumor, and then GL1 makes sure the immune system is ready to jump in and finish the job.
That's a great way to put it. And there's even some evidence that GL1 might actually help the immune system remember those cancer cells that were killed by the chemo so it can recognize and attack them if they ever try to come back.
So it's like training the immune system to be even more vigilant even after the initial treatment is over.
Exactly. And that's why these combination therapies are so exciting. They really hold the potential for achieving more durable responses, maybe even cures, for cancers that were once considered, well, incurable.
That's amazing. But, we've got to be realistic, too. There are always potential downsides with any new therapy, right?
Yeah.
We talked earlier about the risk of overstimulating the immune system.
Oh, absolutely. That's always a concern with any immunotherapy. It's like walking a tightrope, You want to activate the immune response enough to fight the cancer, but you don't want to trigger an autoimmune reaction where the immune system starts attacking healthy tissues.
Right. Too much of a good thing can backfire. So how are researchers addressing that risk?
Well, there's a lot of research focused on identifying those biomarkers we talked about, those clues that can help predict who will respond well to treatment and who might be at higher risk for side effects. And as we learn more about the immune system and how it interacts with cancer, we'll get better and better at tailoring these treatments to each individual patient.
So it's all about personalized medicine, finding the right approach for each person.
Precisely. And that's what makes this whole field so exciting. The possibilities are just incredible.
I agree. This has been a really fascinating deep dive into GL1 and its potential to change the landscape of cancer treatment. We've learned how it works, the cancers it might be most effective against, and even its potential in combination with other therapies. But, there's another layer to all of this that we haven't touched on yet.
Oh, and what's that?
Well, we've talked a lot about the science, but there's also a human side to this story. After the break, we'll explore the personal stories of patients whose lives have been touched by cancer immunotherapy and how these advancements are impacting our lives and our society. We're back. And, we've been digging deep into CD86 and its role in cancer immunotherapy.
Yeah, it's really amazing science, but it's also important to, step back and think about the bigger picture.
Absolutely. We've covered a lot of ground, but sometimes the most powerful stories come from the patients themselves.
That's so true. The human side of this research is really what drives it all.
Exactly. So have you seen firsthand how these therapies are actually impacting people's lives?
Oh, absolutely. I've been lucky enough to work with some incredible patients who have benefited from immunotherapy. And their stories are, well, they're really inspiring.
I bet. Would you be willing to share one of those stories with us?
Sure, yeah. There was this one patient who was diagnosed with advanced melanoma a few years ago.
Melanoma, that's a tough one, right?
It is. And at the time, it was considered really aggressive, really hard to treat. He'd tried all the conventional treatments, but nothing seemed to be working.
So his prognosis wasn't good.
Not really, no. But then he got a chance to enroll in a clinical trial for a new immunotherapy drug. It was targeting CTLA4, one of the receptors we've been talking about.
So it was similar to GL1 in that it was what, trying to release those brakes on the immune system?
Yeah, exactly. And well, the results were pretty amazing. Within a few months, his tumors started to shrink. He had more energy, just a much better quality of life overall.
Wow, that's incredible. It sounds like it was a real turning point for him.
It was. And while not everyone has such a dramatic response, his story really shows us what's possible with these new therapies. They really have the potential to change people's lives.
It also reminds us how important it is to keep pushing forward with research, to keep looking for new breakthroughs.
Absolutely. There's still so much we don't know about the immune system and how it works, but with every new discovery, we get closer to a future where cancer is no longer a death sentence, but something that can be managed, even cured.
I hope so.
Yeah.
But, as we develop these new treatments, we also have to make sure they're accessible to everyone who needs them.
Right, exactly. These therapies can be very expensive, and not everyone has access to them. We need to work on making sure everyone has a fair shot at getting the treatment they need, no matter their financial situation or where they live.
That's such an important point. It's not enough to just develop these amazing treatments. We also have to make sure they reach the people who need them. And I think it's also important for patients to be informed, to understand their options, and feel empowered to make decisions about their own care.
Absolutely. Open communication between patients and their doctors is essential. Patients need to feel comfortable asking questions and advocating for themselves.
That's so true. It's a partnership. Doctors and patients working together to find the best path forward.
Exactly. It's about more than just treating the disease. It's about caring for the whole person.
Well, this has been a truly fascinating conversation. It's incredible to see how far we've come in the fight against cancer, and I'm really excited to see what the future holds.
I am too. There's so much potential, so much hope on the horizon. It's an amazing time to be involved in this field.
I agree. Well, thank you so much for sharing your insights and expertise with us today. This has been a truly illuminating deep dive into the world of CD86 and cancer immunotherapy.
It's been my pleasure. Thanks for having me.
And to all our listeners, thank you for joining us. We hope you've learned something new and maybe even feel a little bit more hopeful about the future of cancer treatment. We'll see you next time.